Hello everyone. Welcome back to our molecular biology lecture series. In this lecture, we're going to continue covering molecular cloning methods. We'll talk about cDNA cloning, I'll introduce PCR, the polymerase chain reaction, and we'll discuss methods for expressing cloned genes. In the last lecture, we had discussed cloning vectors, getting into examples of plasmids, phage, and phage-derived vectors, like cosmids. I explained that through cloning, you can generate different types of libraries. We can build a genomic library, which, as we discussed, is derived from the total genomic DNA of an organism fragmented into pieces. A genomic library is a collection of clones that, together, cover the entire sequence of an organism's genome, with many clones overlapping in regions of sequence. Genomic libraries are commonly used for sequencing applications, playing an important role in deciphering the genome sequences of different organisms. Another kind of DNA library that we can produce through cloning is cDNA libraries. cDNA libraries are made not from an organism's genome, but from its transcriptome, that is, the total mRNA produced from gene transcription. cDNA is complementary DNA that is reverse transcribed from that RNA. Rather than including all of the genomic DNA, a cDNA library only contains genes that are being expressed, and furthermore, it only contains the sequence from those genes that end up in mature mRNA transcripts. The pattern of genes expressed could be specific to a certain cell type or a certain tissue of an organism in a given condition. Here are some examples of why you might want to clone cDNA, reverse transcribed from mRNA, rather than clone the genes as they are on DNA. The gene coding cystic fibrosis transmembrane conductance regulator, or CFTR, is an example of a very long gene on the DNA level that has multiple introns. The gene itself is 230,000 bases with 26 introns, but after splicing, the mRNA is 4,800 bases, and the protein is about 1,500 amino acids. 2% of the primary transcript is retained on the mature mRNA. By cloning only the cDNA, you get rid of most of the non-coding DNA that makes up the majority of the gene. Another example is the dystrophin protein coding gene, which is 2.4 million base pairs long with 78 large intron sequences. At the DNA level, you have coding sequences which are broken up by non-coding sequences that don't get translated into protein. Yet, once that is made into mRNA, these introns get processed and spliced out. A lot of the DNA in genes does not get translated into proteins, and this is common in eukaryotes. It's something we'll come back to in later chapters. So, by making cDNA from mature mRNA, you get rid of most of the sequences that are not translated into proteins. How do you make a cDNA library? To do this, you first extract the RNA from cells. You get the total RNA, and not all of this is mRNA. But in eukaryotes, the mRNA is specifically modified at the 3' end with poly A tails. What you can do is, you can specifically make DNA from mRNA using reverse transcriptase. Reverse transcriptase makes DNA from an RNA template. These reverse transcriptases originated from retroviruses. I briefly talked about viruses that carry their genomes as RNA. Some of these viruses use reverse transcriptase to make DNA, which they can insert into a host genome. We can use reverse transcriptase as a tool to make DNA from the mRNA that we've extracted from a cell. How do you make cDNA specifically from mRNA versus the other kinds of RNA in the cell extract? The reverse transcriptase needs a primer. What kind of primer might specifically anneal to mRNA? A primer that will anneal specifically to the 3' poly A tails of mRNA would be a synthetic single-stranded DNA oligonucleotide consisting of a string of Ts, or an oligo-DT primer. With the oligo-DT primer annealed, you can use the reverse transcriptase enzyme to polymerize the complementary strand of DNA from the mRNA template. Much like DNA template-dependent DNA polymerases, reverse transcriptase synthesizes DNA 5' to 3', but it uses an RNA template. The resulting single-stranded cDNA is hybridized to the mRNA template after reverse transcription. Once you have one strand of your DNA synthesized, you have to make the other strand if you want double-stranded DNA. In order to do that, you need to get rid of this RNA and replace it with DNA. You would use another enzyme, RNase H, and that specifically recognizes a hybrid DNA-RNA double strand and degrades the RNA. If you do a partial treatment with RNase H, it will degrade some of the RNA and it will leave the DNA intact. The remaining fragments of hybridized RNA can act as primers for DNA synthesis using a DNA polymerase. If you use DNA polymerase 1, the 5' exonuclease activity will allow it to degrade a nucleic acid strand upstream of the one it's synthesizing, and this way you can replace all of the RNA with DNA. In this entire process, the RNA gets replaced and then you have a double-stranded DNA that you can clone into a plasmid. In order to clone it into a plasmid, you need to have overhanging ends. You can produce 3' overhangs by using an enzyme called terminal transferase that adds a series of bases to the 3' ends of DNA from deoxyribonucleoside triphosphates. You can use this to add 3' overhangs consisting of a repeat of C's, and so you could do the same thing to the corresponding vector, but instead you add an overhang consisting of a repeat of G's. The overhang of C's on the cDNA is complementary with the overhang of G's on the vector, and you can ligate the two together. Shown here is only one plasmid, but remember you have a pool of RNA from your cell extract, so you end up with many of these plasmids with many different cDNAs in them. You take those and transform your E. coli, and then within the different colonies that grow, you will have different sequences that make up the library, and collectively you have the sequences derived from many different mRNAs that were transcribed in that sample. Often this process of cloning cDNA is not perfect. You may not get the full-length cDNA copy of the mRNA. Shown here on this slide is a method to fix that, to fill in the missing DNA. It's called rapid amplification of cDNA ends, or RACE. Let's say you cloned your cDNA, and from your library you found the clone with the cDNA you were looking for, but it is missing a large piece of the gene because the reverse transcriptase did not finish copying the sequence. What you can do is take the incomplete cDNA from your clone, cut it out of the plasmid, and hybridize the complementary strand to the mRNA that you'd extracted from cells. The DNA will specifically anneal to its complement mRNA, and it can be used as a primer for reverse transcriptase to fill in the missing sequence, templated by the full-length mRNA. Once you've done that, you can add a tail of bases, C's for example, to the 3' end using the terminal transferase enzyme, and then degrade the RNA in this hybrid using RNase H. A DNA oligonucleotide consisting of repeating G's can serve as a primer that anneals to the 3' tail of repeating C's. And using DNA polymerase, you can synthesize the second strand of DNA, giving you double-stranded cDNA. This double-stranded cDNA can then be amplified by polymerase chain reaction, or PCR. For PCR, you need two primers complementary to opposite strands of the template. In this case, your forward primer is the DNA oligonucleotide consisting of repeating G's. And here, the reverse primer is an oligonucleotide with a sequence complementary to the 3' end of the mRNA, which you ought to know from sequencing your original clone, which included the incomplete cDNA. PCR can produce many copies of the cDNA that you can then clone. I'll take a moment here to talk more about PCR. PCR is the polymerase chain reaction. It is a powerful method to amplify specific pieces of DNA. The PCR method was developed by Kary Mullis, who recently passed away in 2019. In 1993, he was awarded the Nobel Prize in Chemistry for his work on PCR, and ever since it was developed, it has been one of the most important tools in molecular biology. The basis of PCR is to use DNA polymerase to specifically amplify a defined sequence of DNA that is determined by a pair of single-stranded DNA oligonucleotide primers that anneal to opposing strands of the double-stranded template. The double-stranded DNA first needs to be denatured, or melted, into separate strands so that the DNA primers can hybridize to them. And this is done by heating the DNA above its melting temperature, or TM, if you will recall from our first lecture. Heating is usually done above 90 degrees Celsius, so it is important to use DNA polymerases that are stable at high temperature. After the template DNA is melted into separate strands, in order for the primers to anneal, you need to lower the temperature so that base pairing can occur. Once the primers are hybridized, you can increase the temperature moderately so that the thermostable DNA polymerase is active and can extend the DNA. You can go through this cycle again and again, and after multiple cycles, you will have exponential amplification of DNA of a very specific length determined by the primers. Why is this? In every subsequent cycle, each strand of copied DNA serves as a template to which the complementary primer can bind. Elongation of the primer can only continue as long as the template allows. Every copied strand begins from a primer, so this limits how long it can be a template for synthesis of the complementary strand. And thus, you amplify DNA molecules of a specific sequence and a specific length. Before the development of modern-day PCR, thermostable DNA polymerases were not available, so for each cycle, you had to keep adding new enzyme because in the previous step, it would have been cooked above 90 degrees Celsius and lost its activity. But now we can use heat-resistant thermostable DNA polymerases. The first such enzyme used for PCR comes from Thermus aquaticus, a bacterium that lives in hot springs. The thermostable DNA polymerase is one important component of PCR. You also need to have template DNA. You need to have a pair of primers that each bind a specific region of the template DNA on opposing strands. And you need to have the building blocks, deoxyribonucleoside triphosphates, or DNTPs. There are three main steps in the PCR cycle. The first step is denaturation of the DNA template. This is done above the melting temperature, so the sample is hot. The second step is to anneal the primers to the template. This is done below the melting temperature, so the sample is cooled. The third step is elongation, and during this step, the sample is warmed to the temperature at which DNA polymerase is most active. There can be some modifications made to the primers that are used for PCR. The sequence at the 3' end of a primer has to be complementary to the template, but the 5' end can have modifications, including a restriction enzyme recognition site. Why is it strictly required that the primer is complementary to the template at the 3' end? DNA polymerase synthesizes DNA 5' to 3'. It can only add bases to the 3' end, so it needs the 3' end of the primer base paired to the template so that bases can be added to the primer in a templated fashion. If you wanted to clone some piece of DNA into a vector, let's say some gene you want to clone, if you were to rely on the native restriction sites, you would have to be very lucky to find the correct sites on the DNA that you want to clone that are also on the vector. Instead, what you could do is you could design primers that anneal to the beginning and the end of the gene at opposing strands. The bases towards the 3' ends of these primers need to be complementary to the template DNA, but the bases towards the 5' ends don't need to. And there you could engineer recognition sites for restriction enzymes at the ends of the primers as long as you make sure that those sites don't occur within the gene that you're trying to clone. PCR will result in amplified DNA fragments with engineered restriction sites flanking the gene that you want to clone. You can then cut the PCR fragment using those restriction enzymes, giving you sticky ends, and you can cut the plasmid vector with enzymes that give compatible sticky ends. And then you can ligate the PCR fragment containing the gene into the vector. You can do the same thing for cloning cDNA of a specific transcript. It's the same principle. You just need to use reverse transcriptase in the initial step to make the DNA from the mRNA sequence. Then you can carry on to do PCR to amplify DNA that you want to clone into a plasmid vector. So instead of making a cDNA library with many different sequences, if you want to clone the cDNA of a single specific transcript from one gene for which you know the sequence, you can design a pair of primers with modifications at the 5' ends that include restriction sites. First, the antisense primer, labeled here as the reverse primer, anneals to the mRNA at its 3' end. Single-stranded cDNA, complementary to the mRNA, is synthesized by reverse transcriptase. The sense primer, or the forward primer as it's labeled here, can then anneal to the newly synthesized antisense DNA strand, and DNA polymerase can synthesize the second strand of DNA elongated from this primer. Both forward and reverse primers have restriction sites engineered into their 5' ends. The resulting double-stranded DNA can be amplified by PCR, and the PCR product will have recognition sites flanking the cDNA, which can be cut with restriction enzymes, giving a fragment with sticky ends that can be ligated into a plasmid vector cut with the same enzymes. We've seen so far that PCR can be very useful for amplifying copies of DNA fragments that you may wish to clone. PCR can also be very useful in detecting very small amounts of DNA or RNA, because it can be used to amplify minute and otherwise indetectably small amounts of nucleic acids to levels that can be measured. Shown here is one such application of PCR. This is real-time quantitative PCR. It's a method for measuring the amount of a specific DNA or RNA sequence in a sample. In this PCR experiment, you have a fluorescent signal released at each cycle of amplification that is proportional to the amount of DNA that is copied. This particular example of real-time quantitative PCR relies upon the 5' to 3' exonuclease activity of a DNA polymerase to cleave a reporter group that you also have in this experiment, to release fluorescence each time it copies a specific sequence of DNA. In addition to the pair of oligonucleotide primers that you have in a typical PCR experiment, you have a third specialized oligonucleotide, acting as a reporter probe that hybridizes in the middle of the sequence that will be amplified. It has a fluorescent tag attached on one end and a quencher attached to the other. Having the quencher attached to the same DNA oligonucleotide prevents the fluorescent tag from giving off fluorescence. But during each cycle of PCR amplification, as the DNA polymerase extends the primer that binds the same strand as the probe, it will cleave the probe through its 5' to 3' exonuclease activity, releasing the fluorescent tag from the quencher, resulting in a fluorescent signal. Initially, in the first PCR cycles, this signal may be too low to measure, but with every cycle as DNA is amplified exponentially, the signal increases exponentially until it is measurable. This way, you can detect and quantify very minuscule amounts of DNA. You can also quantify RNA if you first reverse transcribe the RNA before carrying out PCR. This can be very useful in measuring levels of gene transcription. It can tell you accurately how actively a gene is being transcribed by measuring the amount of mRNA from a sample. Now that I've introduced PCR and its uses for cloning DNA, along with its applications in measuring DNA and RNA, I will pause here before moving on to discuss methods for expressing cloned genes in the next part of the lecture.